we first need to acknowledge that today biology is foremost an information science. And what do I mean by that? There has been more than 80 years of innovation that led us to the point where we actually talk about biology as an information science. It started with the discovery of the DNA, its structure, evidence that an information flow via the central dogma of molecular biology that provides us with fundamental framework for understanding how genetic information flows and proceeds to create functional parts that we can consider as parts of the biological system. And it helped us to establish the link between the organism's genetic makeup, so the genotype, and its observable characteristic, or the traits, so phenotype. But we knew that this simplistic linear model doesn't always work, and for most, it led us to a philosophical shift. For a long time, we've been conducting our research following so-called reductionist approach. So we assume that by knowing all building blocks of a system, we will fully understand how the system works. But it was already the Aristotle that said that the whole is greater than the sum of its parts. So in the last 25 years or so, we have the emergence of a field called systems biology, where we're actually interested in how the components of a biological system interact, either with one another or with their environment, and how the emerging behavior occurs. And then we can produce the system's overall overview. So in my group, located in Aachen, at the border of 3 countries, you can see here a picture of my team standing between the Netherlands, Belgium, and Germany. We're actually capitalizing on this transformation and this shift. We're using mathematical modeling and data-driven approaches to unravel different biological processes. But I was asked to maybe mention a few words. How did I find myself leading such a group? What was my story? How did I get there? So hopefully not boring you. My path, my scientific path, was far from being linear. Although I already specialized in so-called biomathematics at Politechnika Gdańska, my first job out of the university was in the financial sector. I was building cost allocation models, working with the SQL database, doing nothing related with biology. And due to personal shifts, I moved to Scotland and went back to school to earn my second degree from School of Medicine of the University of Aberdeen. Here you can see a picture of me proudly presented with my parents at the graduation ceremony. And at that point, I was super oriented at working in the R&D sector of a big pharma to provide computer-supported design for new drug targets. My study coordinator advised me that in order to have a leading position in R&D in such a companies, I need to have a PhD. So I started to look for computational PhDs, but at that time, the only interesting PhD I found in Aberdeen was with plants. So after a lot of discussions with people much smarter than me, I was convinced, well, modeling is modeling. If you understand how to do it in one domain, you will know how to transfer it with the other. Let's do it. I started my PhD in Aberdeen, working in a project called ACLIFOT. Here you have a group of all super motivated PhD students in a white lab coat. It was probably somewhere around first 6 months. I was attending a conference in France that was a students-driven meeting on Chlamydomonas. And there I realized there is no coming back. I want to be the one that will work on issues related with how to feed people and how to understand plants' growth, rather than how to medicate them. And then I moved to Germany during my PhD time. I graduated in 2016 from the University of Heinrich Heine in Düsseldorf. I got trained in quantitative and theoretical biology, but also in experimental spectrographic techniques. I was performing my own experiments in Switzerland, France, and Japan. And first years as a postdoc were divided between finding myself in the role of a mother and conceptualizing my own line of research to get my own independent research funds. In contrast to what I remember about the academic system in Poland, in Germany, it is really expected that a scientist move in between the institutions before they get tenured. Therefore, after a PhD, if you're good and you really do plan a career in research, it is expected you will move somewhere else for postdoc. Having a small kid at home and just actually, starting the COVID pandemic, I realized I don't want to drop my network right now. The only way to stay in the same place is to actually get my own research funding, which I managed to get. And in 2019, I started my first individual research project working with tomatoes. Within 4.5 years after my PhD defense, I received a call at RWTH Aachen to start my own research and own independent group as a W1 or junior professor. And from then, my path started to be more linear, as you can see on this side. For the past 4 years, I focused more on acquiring funds, building the intellectual capacity, recruiting people, writing papers. And one of the currently being paper that we're writing, I would like to present to you today in the scientific part of this talk. So moving to the scientific part, I want you all to think for 3 seconds. What is the first thing that comes to your mind when I say model? Because I have used that word already several times. And be honest, because when my students are honest, they admit that this is the very first thing that comes to their mind. But I would expect that in this group that is sitting now in Lublin, some of you have been thinking about your favorite model organism. And whatever was in your mind, it was a simplified representation of a much more complex, of a much more complex system. And in our work, we also do not operate only with one type of a model. If you're going to take a look at this cycle of model buildup and creation, you're going to see that I use the word model 3 times. We construct a conceptual model that gets formalized in mathematical terms that then gets programmed into computational model. But very importantly, all of our models start with a real life question that quite often is way too ambitious and way too broad to be addressed by one person or one research group. Therefore, we need to specify it. So instead of asking, how can we have plants that withstand the climate changes? We specify how can we have a plant that withstand changes into temperature. And then we conceptualize the model by thinking, what do we focus on first? A whole system or just roots or just leaves? What do we assume will be constant over a time, even though we know there might be some dynamic changes? What are our boundaries of the system? And once we have the concept, we start to formalize it. And there's numerous ways how we can build the mathematical models. The most popular classification that you might have heard of is division of the models into the so-called white and black boxes. We live in the revolutionary times of an emergence of highly powerful algorithms that provide high predictive power. And therefore, we have an emergence of black box models where we don't necessarily know what is happening inside, but by providing a high quality input, we can have a high quality output. In contrast to this type of models, we had those white box models that are built on a first principles where they are highly interpretable, but they are often incomplete or computationally expensive. Therefore, in my lab, we're actually merging those 2 types of models in a so-called hybrid modeling. And there we can combine the predictive accuracy of the black box models and machine learning with the mechanistic insight, keeping them robust. And one process that we investigate from very different angles in the lab is photosynthesis. We don't investigate it in vitro or in vivo, but in silico. So few words why anyone is bothered with photosynthesis today. I like to use this argument that if we think about the food production today, considering the predictions about the amount of people being on this earth in future, this would be the amount of food that we need to produce in 2050. And the challenge itself is not to produce it, but to consider that we don't have additional land anymore, that we have already today limited water resources, and whatever actions we're going to take, we need to maintain biodiversity and reduce our environmental impact. And so, after many years of targeted research, it has been proposed, why not to improve photosynthesis, the very beginning of plant growth. Can we consider photosynthesis as an innovative target for improving crop production? And so, in our lab, we indeed try to understand a lot of dynamic processes that are affecting the efficiency of the photosynthetic process, but we also are interested in photosynthetic organisms in the context of their biotechnological exploitation. What do we need to change in the system, either on a genetic level or on the environmental growth level, in order to, for example, increase the secondary metabolism production? To maybe your surprise, we actually are using also photosynthesis as our educational process. If you have your phones, I don't see you actually, so it's very weird, but if you have your phones, you can scan this QR code and it will take you to an online tool that we released at the beginning of this year that will teach you how you can use modeling in the context of photosynthesis research. And what I really appreciate about this design is that you can select whether you come more from a biological or mathematical background, so the context of the website will be changed accordingly, providing more details on one side or the other. So there is plenty of projects running at the moment, and I thought we will have a deeper dive into one particular project, which once again was completely not linear. This work is a result of a collaboration between our computational lab and my PhD student Tobias Fenning and the experimental group of Ilka Axmann in Düsseldorf. So why was that collaboration not straightforward? Because Tobias was initially interested in rice, while Ilka is an expert in cyanobacteria. So let's dive into it. When Tobias started his PhD, he was interested in improving rice production, and he came across this publication from 2020 where roots of rice were inoculated with a nitrogen-fixing cyanobacteria that improved the overall growth of rice. And then Tobias started to look through pictures of rice fields. And if you notice here, the rice is grown in an aquatic area. So Tobias came back with a question. If the cyanobacteria are in this environment, do they still perceive light? And does it mean that we can assume that they're photosynthetically active? And then in terms of light, they will definitely not see the same light as we outside. You can see the spectra of the solar light on this graph because they're going to be on a different depth and there is a different scattering of light and different absorption through the pigments. So the spectra of the light that they're going to be exposed to will be significantly different. And that's how Tobias' very initial project has shifted its focus. Tobias was committed to understand what is the impact of light spectrum and not only intensity on photosynthetic performance. And because I know I'm talking to the young researchers, I wanted to highlight here that sometimes the project will go in a completely different direction, and it's worth to explore those areas where you clearly identify a gap of knowledge, even if that was not necessarily the first thing that you noted down in your lab book as the goal for the next 4 years. So what we started to do resembles a little bit like playing with puzzles. You go through tons of literature and you try to find whatever is there described about the cyanobacteria, about the model organisms that you work with. You need information about the protein concentration. You need information about the pigment absorption. You need information about some measured electron transfer rates. And whatever you take, you try to put together into the system picture in order to make a coherent picture of an organism. Now, we wanted to understand how the cyanobacteria work under different light regimes. Therefore, we ask our experimental collaborators what are the light spectra of the light sources that they use in the lab. And you can see here how much do they differ if someone is using the white LED but some groups are using a cool white LED with a high peak around blue light, and other groups are using warm white LED with a much broader coverage in the red region. In order to have a model that can capture the differences of the different light spectra, we needed to consider that each of the pigments inside of the organism perceive the light differently. And you can see here, we selected 4 different pigments that have been named on the top, and they are absorbing the light of a different spectra. We're considering the light absorbed by each pigment by multiplying the product of the absorption with the effective light intensity in a culture and integrated with the Lambert-Beer function. That allowed us to calculate not only for individual monochromatic light, but for any combination of light. Now, what you can see on this slide is our conceptual model of what we decided to focus on when we wanted to understand the photosynthetic dynamics of a cyanobacteria. You can see here the picture of the protein photosystem 2 and some other protein complexes, electron carriers, etc. I don't think we have enough time to dive in. So the important part is that after finishing this conceptual work, we translated it into the mathematical formalism. We use differential equations because they allow us to monitor what is the rate of change over the time in those pigment complexes and the redox states of different electron carriers. Now, when we were going through our puzzling work, we were searching for evidence that some of the reactions are, for example, catalyzed by an enzyme. If we didn't find the information about the catalysis, we were assuming that the reaction will be linearly dependent on the concentration of the substrate. And we call it then that it's represented with a mass action kinetics. Then, depending on the evidence that we were finding, sometimes we were learning that reactions are, for example, reversible, but only under certain environmental conditions. So we always also needed to incorporate that information. Once we had our system, we were capable of, for example, simulating the distribution between the different electron flows. Now, why is it important? You might have heard that for years there is an interest in using photosynthetic microorganisms for biofuel production, including, for example, hydrogen production. All of those biofuels will be produced from the energy that is either produced through the linear electron flow or is being manipulated by the division between the different other alternative pathways, like, for example, cyclic. So to understand in which ratio they operate under which conditions and how to manipulate it to increase, for example, the green flow versus the blue flow is of a high relevance for the biotechnological groups. And so to answer the very first initial question, so what is the effect of light spectra on the growth of the cells? We run this simulation. You have 4 sources of light, as presented a few slides ago. We run our simulation up until they reached a steady state for various light intensities and read out the steady state of the carbon fixation. And we've noticed that this discrepancy between cool white LED and warm white LED is massive, because although you're going to get to this eventually to the same carbon fixation rate, you can use half of the light intensity if you're going to use the warm white LED, while you're going to have to use 600 micromole of photons per meter square per second with a cool white LED, which translates to energy consumption. So this slide convinces always our collaborators when we ask them, what is your light sources? How do you grow your plants? How do you grow your bacteria? There is a difference, even if you tell us white LED, show us the spectra. What else can we do? We can simulate what would be the production under monochromatic light and also see under which light intensity will the monochromatic light lead to the same output. But let's go back to Ilka. What about her? Well, we wanted to understand how a unicellular aquatic organism perceives different light intensities. And therefore, we focus on a fluctuating light and spectrodependent model. What Ilka was interested, even though she was working with a photosynthetic organism, she was interested in the secondary metabolites that can be produced by the photosynthetic organisms. You might be aware that as a community, we identified so far more than 200,000 plant secondary metabolites. And it's good to know that cyanobacteria can also produce many of them. So Ilka wanted to understand, can she increase the production of terpenoids in a cyanobacteria? And a particular interest of her lab was squalene, and that's how we come to the shark. The squalene is a linear polyunsaturated triterpene, and it has a lot of functions. It is used in the skincare because it's an antioxidant. And a few years back, there was an increased demand for squalene because it was used as a stabilizer in a COVID-19 vaccines. Now, the problem is that one of the main natural resources of a squalene is the shark liver. So when the demand increases, there's naturally an increase for a high production that will be more ethical and more sustainable. Hence, cyanobacteria. But now let's take a look at this MAP pathway. If you're gonna carefully look, the squalenes are here. And here we have carotenoids and chlorophylls, which means that the synthesis of the pigments that absorb light and drive photosynthesis for the production of sugars that sustain the growth of the organisms are produced exactly from the same precursors as the squalene. So a natural question was, is there a theoretical maximum by how much can we increase the squalene production without inhibiting the cell growth? And we got Andreas Nakielski on board, who beautifully merged the methods from both labs, theoretical and experimental. And one of the first things he did was to show to us that actually the growth rate is dependent on the chlorophyll synthesis rate. So even when he managed to have a pigment, use some pigment inhibitors and have less of the pigment concentration, the cells still maintain their growth rate. He built a model of the MEP pathway that had 21 enzymatic reactions and could simulate what is the concentration of each metabolite at different time steps, and performed proteomic and metabolomic analysis in order to calibrate and feed the model, and then wanted to merge our previously developed model for the photosynthesis of cyanobacteria to take into account that at every iteration, when we're going to have less carotenoids or less chlorophylls, we will produce less ATP or NADPH. But the model that I just presented to you, Tobias' model, was quite big, 17 differential equations, a lot of parameters. That was a bit of an overkill to merge those two things, especially that they operated on a different time scale. Those of you that are better biochemists than I am will know that the pigment synthesis takes hours, and the dynamics of the photosynthetic system is on a scale of sometimes pico-nanoseconds, seconds, maybe minutes. So here we go to our hybrid approach. We produce synthetic data, we assume that there's different light intensities and different pigment compositions, and run our model hundreds of times to produce the output, what would be the energy production under the given inputs. And with this huge set of data that we generated, we could surrogate our model, basically approximate it with a much simpler function, which was a 7th degree polynomial. And now, because I present to you an ongoing research, you can see the current state of the model. With the blue line, you can see that we can quite well reproduce the experimental cell density for the first day and a half. But for a longer time, we actually overestimate the cell density, meaning that we are missing still some sort of an inhibitory process, and we are at the stage of investigating it. And with that, I wanted to show you just one last slide of what we can do theoretically before the experimentals get to test it. There is a method called metabolic control analysis that allowed us to, for example, theoretically run overexpression experiments and find out how many genes do we need to overexpress in order to lead to the increased squalene production. And from our first test, we could find that by only overexpressing 4 genes that are named here, we could theoretically increase the squalene production by 10-fold. And with that, I wanted to thank you for your attention. I wish you a very successful 2 days of a workshop and a lot of success for your PhD.